Welcome to the session of somatic receptors and special senses. And in this session, our learning outcomes would be at the level of classification and description of the receptors, both in the body wall as well as in the organs, and belonging to the general domain as well as belonging to the domain of the special senses. So before we move on to the classification and the learning outcomes, it would be useful to think of the function of the receptors or what they are actually doing for the animal. So they are basically helping the animal communicate with its environment. The environment can be internal or it could be external. And if we look at the function or how they work, I think it would be useful to think of a physics concept where you studied the transducers. And if you remember that transducers, typically they convert one form of energy into another form. So if you think of a transducer and compare it to receptor, you will find many similarities. Because I will just give you two examples. One of our receptor is the nasal epithelium, the olfactory epithelium, for the sense of smell. So the receptors which are there to detect the sense of smell, they are basically converting chemical energy, which is the composition of the molecules which are giving a particular smell. So they are converting that chemical form of energy into electrical form, which is the impulse generated in the olfactory nerve. Or you could think of the thermal receptors. These are the receptors which are sensitive to temperature and are helping. They are basically the exteroceptors, that is, they are external in location. And if you think of the thermoreceptors, they are basically converting the heat energy into the electrical energy. So if this is how you think of a receptor, it will help you understand more about their function, and that would also help you remember their structures which are involved with the whole process of nerve impulse generation. So all these receptors, unless they are nerve endings or the simple nerve endings, they usually are formed of multiple cells and associated structures, which can be supporting structures or which can also be the nerves which are emerging from these receptors, the sensory nerves. So having understood how they basically work, that is like a transducer, we now move on to the subdivisions of the receptor. We also call it as the subdivision of the afferent system. Afferent because this is sensory in nature. So we have one bigger subdivision of afferent system, that is the general afferent system. And the term general tells you that they are associated with the general sensations of pressure, pain, stretch, or temperature. This pressure is also, it's a extreme form of the sensation of touch. So these are the basic sensations which are dealt with the general afferents. Then the other bigger subdivision is the special afferent group. And the special afferent receptors, or special afferents as we call them, they deal with the sense of vision, sense of taste, smell, audition, that is hearing, and balance. So if you look at this 1, 2, 3, 4, and 5 senses, sometimes the last two are collected or dealt with together. So these 4 or 5 senses are the senses we also called as our special senses. So the afferent system of the animal's body, which is dealing with these senses, is called as the special afferent. So these are the two major divisions. I would again repeat. The general afferent is distributed throughout the body, and they are associated with the general sensations of pressure. It could be anywhere, pain, it could be anywhere, stretch, and temperature. Whereas the special afferent system is very localized and is present in the eye, present in the ear, present in the nose, and in the oral cavity. So this is more special in its function. Now looking at the general afferent, it again has two divisions. One general afferent is general somatic afferent, and as the name indicates, it is associated with the general body wall. Soma is the term which is used for body wall. So general somatic afferent, the sensory system which is associated with the body wall of the animal. And within this system, there are two types of receptors that you can find. One are the exteroceptors, and these are the receptors which are helping the animal communicate with its external environment. That's why we call it as exteroceptors. And they receive the external stimuli of pressure or touch or pain or stretch or temperature in the term of cold and heat. So different receptors are there for each of them, and they are grouped as exteroceptors. The second category within GSA or general somatic afferent is the proprioceptors. And these are the receptors which are sensitive to stretch. And you find them in the muscles, you find them in the joint capsule. We will, if you remember, we once talked about when we were discussing myology, our Golgi bodies and the muscular, muscle spindles. They are the proprioceptors. And the nerve fibers for these receptors, the GSAs, they are present in all spinal nerves. All spinal nerve carry sensory fibers which enter into the spinal cord, into the dorsal root, and they are also present in the cranial nerve number 5, which is the trigeminal nerve. So these are the nerves which will be carrying the GSA fibers. Then within the general afferents, our next subdivision is the general visceral afferents. And the name should indicate that these afferents or sensory endings or receptors, they are present in the organs or within the viscera. They are basically free nerve endings, and they are also called as nociceptors, or they are sensitive to pain. You can find them in blood vessels, you can find them in glands and viscera. And in addition to that, in organ pain, they are also sensitive to stretch, which if it is excessive may result in pain, or they are also sensitive to changes in the chemical composition of the fluids around them. So these are the GVAs. And the fibers for GVAs, they are present in all spinal nerves, and additionally in the cranial nerve number 3, which is oculomotor, 5, which is trigeminal, 7, which is facial, 9, which is glossopharyngeal, and 10, which is vagus. That only tells us that these cranial nerves, they have sensory function associated with them. You can pause here and just review the information that we have shared so far about the general afferents. And once you are done with that revision, then you can continue with the recording and listen to the special afferents. Now within the special afferents, we have again two groups, and the first one is the special somatic afferent. So special, we already know, is associated with certain special senses, and somatic mean body wall. So those special senses in the animals whose receptors are present in the body wall, or they have ectodermal origin part of them, so they are included in this one. And they are the eye, retina, and the ear in the form of vestibulocochlear organ. And the sensory fibers for these two nerves, if you remember from the last lecture, They are present in cranial nerve number 2, that is optic nerve, and cranial nerve number 8, which is vestibulocochlear or auditory nerve. Special visceral afferent, again related to special organs, but those special senses whose receptors or whose afferents or beginning or starting point is within the organ systems or within the viscera of the animal. And these are two of them. One is the sense of smell, which is the present within the nose, and the other is the sense of taste, which is present in the oral cavity. And the sensory nerves for these afferents, they are present in the cranial nerve number 1, which is olfactory nerve, cranial nerve number 7 and 9, which are facial and glossopharyngeal nerve. So that means that the facial and glossopharyngeal nerve are those cranial nerves which are associated with the sense of taste. Now, this is a general layout of the afferent system of the animal or the receptors which are present throughout its body and what are its subdivisions and where do you find the nerve fibers for those afferents. So now we move on to our general somatic afferents, and these are the cutaneous receptors as we discussed, as well as the proprioceptors. So within the cutaneous receptors, there are possibilities that some of them would be free nerve endings. That is, only a nerve ending would be there without any capsulation or multiple layers covering its terminal end. They are present in the skin also. They are sensitive to pain. We call them nociceptors. Some of them are also sensitive to touch, and even some of them are sensitive to temperature. So they are sensitive to different modalities, as we said, pain or temperature, and sometimes the tactile discs in the form of sensitive to touch. So this is one form of them where the nerve endings structurally have no covering or capsulation on them, and we call them as free nerve endings. The other possibility is that these cutaneous receptors would be encapsulated, that is, that they would be covered with multiple layers. And they can be mechanical receptors, that is, they are sensitive to pressure or touch. Meissner corpuscles are part of them. Pacinian corpuscle are also part of them. Ruffini's are also considered as part of the mechanical receptors. But we also have these end bulb of Krauss. They were considered, I mean, there are two schools of thoughts. One of them says that they are the cold receptors, and there is another school of thought which says that the temperature nerve endings are free nerve endings. But you can get more information about it from your histology teachers. They would be more updated on that what is the consensus about the thermal receptors. Do they have encapsulations, or there are both possibilities that some of them have capsulations and some of them are free endings. Within birds, you see two specific receptors which are not present in other mammals. And these are the Granry's corpuscle and corpuscles of Herbst. They are basically sensitive to pressure, and they are present around the beak of the animal. And when the birds, and when they dip their beak in the water or under the sand, then they are sensitive to the vibration of other insects around that. And that vibrations helps them detect the food which is there. So these are basically sensitive to velocity or the waves which are touching them or colliding with them. In addition to these cutaneous receptor, the other category within the general somatic afferents is that of proprioceptors. And as we discussed, that they are present in the muscles, they are also present in the tendons and in the joint capsules, and they are sensitive to stretch or the weight on the animal at that point. We would rather use the word stretch instead of weight. So two of them are known to us here, more important for us. One is the neuromuscular spindle and other is the Golgi tendon organ. The neuromuscular spindle, They are present in the intrafusal fibers in the skeletal muscles. If this is the whole of the skeletal muscle, then they are regular extrafusal contractile fibers, which is called as the skeletal muscle fiber. And embedded within these typical skeletal muscle fibers are specialized intrafusal fibers. These are also basically skeletal muscle fibers, but they are specialized because they have specialized nerve endings associated with them. And these nerve endings are together making the neuromuscular spindle. So within these intrafusal fibers, associated with these intrafusal fibers, which are enclosed in a sheath, we have certain nerve endings. Among them, some of them are sensory, which we call as proprioceptive nerves. And these are the annulospiral and the flower spray. If you trace them back, annulospiral are the one which are spirally organized around intrafusal fibers, and the flower spray are the ones which are scattering like a tree or a plant over the intrafusal fibers. So these are proprioceptive, or the sensory or the efferents of the neuromuscular spindle. So the neuromuscular spindle also has a motor control, which is provided through the gamma motor neurons. So this is where your proprioception sensation arises. Proprioception, I repeat, is the awareness of the animal of its posture. And how this awareness or adjustment to the posture is made, it is made through these neuromuscular spindles. So basically these neuromuscular spindles help the animal maintain its posture and respond to the stretch which is there on the muscle. And as a response to the stretch, the animal then changes its muscle tone. Either it relaxes the muscle or it contracts the muscle, just to respond to the stimulus. Another component of the proprioceptor is the Golgi tendon organs. The Golgi tendon organs are present at the musculotendinous junction, where the muscle tissue is transforming or transcending into the tendinous tissue. And these are the Golgi tendon organs, sensory nerve endings basically. And the stimulus which travels through them, it goes through to the spinal cord, and there this neuron, which was sensory in nature, it goes and makes a synapse with the inhibitory interneuron. Inhibitory interneuron has an inhibitory effect on the alpha motor neuron, which are innervating the extrafusal fibers. So the process is that when there is a stretch on the muscle, and this stretch is sensed in the Golgi tendon organs, they send some information to the spinal cord, and through the inhibitory neuron, that alpha motor neuron sends the message for the skeletal muscle fibers to relax. You see, it is inhibitory neuron. Inhibitory means it inhibits the contraction of the muscle in simplest form. So through this inhibitory neuron, the action of the alpha motor neuron, it allows the animal to adjust its muscle tone in accordance with the stretch which was sensed at the Golgi tendon organs. So this was briefly the description about the proprioceptors. And the very small mention of the general visceral efferents, we said that these are the sensory nerve endings which are present in the viscera. And we said that they could be present in the glands or in the vessels, and they are sensitive to chemical stimuli, and they are also sensitive to stretch. That they are basically naked endings in the mucosa of the organs. So with this, we achieve our first learning outcome, which was the classification and exemplification of the general somatic receptors. And we very briefly touched upon the general visceral receptors also. With this, we move on to the next learning outcome, that is to be able to describe, draw, and label the structures involved in the sense of vision. So we know that sense of vision, it belongs to our special somatic afferents. And the structures which are working for the sense of vision of the animal are its eyeball, and then the optic nerve, which is formed at the caudal end of the eyeball. And then this optic nerve, it reaches the brain, and the information is processed in the visual area of the cerebral cortex. So these are the nervous components: the retinal layer of the eyeball, the optic nerve, and the visual area of the cerebral cortex. These three neural structures are involved in the image formation. And of course, the whole process is supported by adnexa. Adnexa is the term used for the supporting tissue for the eyeball. We would come back to it. In this diagram, I would just ask you to have a look at it and identify or familiarize yourself with the terms. Tarsal plate. This is a connective tissue structure. And this tarsal plate, it allows the anchoring of the cilia or the eyelashes, and it is associated with glands. We would learn about them, which are called as tarsal glands or the meibomian glands. Then the eyeball is lined by, on the outer side, it is covered by a conjunctiva, which is a very thin transparent layer. And this conjunctiva is not only covering the eyeball, and here we call it as the bulbar conjunctiva, but it reflects on the undersurface of the eyelid, and here we call it as palpebral conjunctiva. You see, so there is a palpebral conjunctiva. Palpebra, the term is used for the eyelids. So the conjunctiva, or the thin layer which is lining the underside of the eyelid, is called as palpebral conjunctiva. Then it reflects on the eyeball, and here we call it as the bulbar conjunctiva. And this small space between the eyelids and the eyeball, or the palpebral conjunctiva and the bulbar conjunctiva, this small space in which we usually administer medicines, is called as conjunctival sac or conjunctival fornix. So then we see what is cornea. Cornea is the transparent layer which is covering the front surface of the eyeball, covering the iris and the lens. And the space which is opening of the eye, this is actually the space between the two eyelids. The slit opening is called as the palpebral fissure, and the rest of the things we have already discussed. So when we are talking about eye, the anatomical terminologies are slightly modified. The term anterior is also used here for rostral. This is only true for eyeball. Generally, we don't use the term anterior within the head. We only use rostral, but with the eyeballs, sometimes the term anterior is also used. Posterior and caudal are also used to indicate the surface which is towards the tail of the animal. Then the lateral surface of the eyeball is also called as temporal surface because it is towards the temporal bone, and the medial surface of the eyeball is also called as the nasal surface or the nasal direction because it is towards the nasal bone. So when we will study the structure of the eyeball, we would be basically focusing on the three layers of the eyeball and the three chambers which are present in addition to the lens. So looking at the layers, the fibrous layer, the vascular layer, and the nervous layer. This is the sequence of the layers from outside inwards. So let's see the first outermost layer, which is called as sclera. This is the white of the eyeball. And the posterior 2/3 or 4/5 is opaque and white in color, this outermost layer, and it is tough and fibrous. It provides a structure or shape or support to the eyeball. But as it comes to the rostral or anterior surface of the eyeball, it becomes transparent, and its cranial or the anterior or the rostral 1/5 or surface, this is called as cornea. This cornea is transparent, and it has no blood supply of its own, and it receives its nutrition from the fluid which is present in the eyeball. And here we have the lacrimal fluid. So I repeat that outermost layer is called as sclera. Its posterior 4/5 is dense, white, opaque. And as you look into the mirror, the white of the eye is the sclera. And just in front of the lens, it's not white, but it's transparent. And this structure is called as the cornea. And this junction is called as the sclerocorneal junction. It is also called as limbus at some points. Then we have the second layer, which is blue in color here, and it is vascular in nature. It is very rich in blood supply. This blue layer, when it is in the caudal part of the eyeball, we call it as the choroid. And as it goes forwards, then it contributes to the formation of the ciliary body. And ciliary body, and then it continues further as the iris of the eye. So iris, which is giving the color to the eyes. Some people have green, some have blue. You see some cats having yellow eyes. So that color is coming from the iris, and it continues caudally, and it is connected to the ciliary body, and then it continues caudally and then connected to the choroid. So these are in continuation with each other. And the ciliary body is that structure which has which is innervated by the third cranial nerve, oculomotor, and it is connected to the lens with the help of the suspensory ligament of the lens. It is also called as zonular fibers. And the contraction of the ciliary body, it results in the accommodation process. That is, it changes the shape of the lens, and as the lens changes its shape or the thickness, it changes the focus of the eye. So the role of the ciliary body is basically with the allowing the animal to have its focus, and it is innervated by the third cranial nerve, which is oculomotor. Then we have the innermost layer, which is the nervous layer and is called as the retina. And this retina, it is sensory, and this is actually that layer of the eyeball which is related to our special somatic afferent, that is special senses neuroepithelium is here. And it is a neuroepithelium, basically neurosensory epithelium, sorry. And this the nerve fibers from the cell bodies in the retina, they actually come together to form the optic nerve. So I repeat here, this optic nerve, which is going to the brain, this is the second cranial nerve. How it is formed, this is basically formed of the axons or the nerve fibers of the nerve cell bodies which are present in the retinal layer. So these are the three layers of the eyeball from outside inwards. Similarly, we have three spaces also, or chambers. This larger space, which is present between the lens and the retina, is called as vitreous body, and it is filled with jelly-like fluid. And so this vitreous body, it gives the eyeball a proper shape. Then we have two more chambers. There is an interior chamber here, this space between the cornea and the iris. And then we have a very narrow, small posterior chamber, and this chamber is between the iris and the lens. And these two chambers are filled with aqueous humor, and this aqueous humor basically is produced by the epithelium here, secretory epithelium, and it releases this aqueous humor here, and it is also reabsorbed back by small venules present here, and therefore there is a constant balance between the production and reabsorption of the aqueous humor. So if there is an imbalance and there is a blockage and there is little reabsorption, that results in increase in the intraocular pressure, and that is known as the glaucoma, which is a pathology here. So these are the three chambers, and you see that they have different functions and different structures filling them up. Then in addition to the eyeball, we also have certain supporting structures with the eye, which support the function of the eye and support the integrity of its tissue. And these are called as adnexa, and they include the orbital space and the fat which is present, the connective tissue layers which are there, the extrinsic muscles of the eyeball, which are 7 in number. 4 of them are called as the straight muscle, medial, lateral, dorsal, and ventral straight muscles. And 2 of them are the oblique muscle, the dorsal and ventral oblique muscles. And one is the retractor oculi muscle, which is present around the optic nerve, which is present caudal to the eyeball. So these 7 muscles, they technically support the movements of the eyeball. When you rotate your eye or when you turn your eyeball to the right or left or up or down, then you're actually using these straight or the oblique muscles for the movement of eyeball. And also, of course, we have associated vessels, nerves, and lymphatics. There's one more structure which is associated with the eye and is helping with the function of the eye, and that is called as nasolacrimal system. We'll have a look on it here. So what nasolacrimal system basically is, it is a system of certain glands which are present around the eyeball, and they produce secretions. These secretions are watery secretions, although they also have certain lipid and mucoid component. And these secretions are for lubrication of the eye, because otherwise it would be too dry. And they are continuously being produced in the conjunctival sac here, and they are continuously being reabsorbed back or exiting the space here. So we will see what these glands are and what they are producing and how they are running. First thing is the lacrimal gland. It is also called as tear gland. As you can see here, it is present laterally and dorsolaterally in relation to the eyeball, and in case of dogs, it is present deep to the supraorbital ligament, which we have here close to the zygomatic arch. So this gland has multiple small lacrimal ducts which are opening into this conjunctival sac or conjunctival fornix, and they are releasing their secretion, which are largely watery secretions. In addition to the secretion of the lacrimal gland, we also have a gland here, gland of the third eyelid. This is important to remember or understand here, that we are typically, we know that there are 2 eyelids. One is the upper eyelid, one is the lower eyelid, or the dorsal or the ventral eyelid. But we also have this nictitating membrane or the third eyelid towards the medial canthus or medial commissure of the eye. This is the medial angle, this is the lateral angle. So near the medial angle, there is this fold of connective tissue, which is vascular and is called as the third eyelid or nictitating membrane. It is also associated with a gland on its bulbar surface, and that also releases secretions here. So secretions of these glands, they are basically drained with the help of this duct system. So we have these 2 small openings. You can also look for these openings in your eyes if you look in front of the mirror, and we call these openings as lacrimal puncta. These openings, they lead to small canaliculi, these very small ducts, which come together to make one single duct, which is called as nasolacrimal duct. And this duct opens into the middle nasal meatus. So if you have ever seen that when your eyes are filled with water or with tears, then there's fluid coming out of the nose also. And this fluid is basically the same fluid which is here. If you have ever felt that if you have eye medicine or eye drops administered in your eyes, Then you also taste them in your throat. Why does it happen? Think about the link that why administering eye drops in your eyeballs will give you a bitter or different taste in your throat. I'll ask this thing on Monday. Anyways, so apart from the lacrimal gland and gland of the third eyelid, we have also some accessory glands here associated. One of them is called as the meibomian or the tarsal glands. And these are the glands associated with the base of the eye, these eyelashes or cilia. And they are releasing some lipid-based secretions. Then we also have glands of Mole and glands of Zeiss. Their function is not too known, but the secretions of all these glands together, it helps in the formation of a fluid layer here, which is called as precorneal film, which prevents the cornea from drying. So this much for our learning outcomes related to the special somatic afferent, particularly for the eye, where we are talking here about the gross anatomy, the histology of the retina, the neurosensory epithelium there, that would be discussed in your histology classes. But here we only discussed the route taken by the nerves there from eye optic nerve to the visual area in the brain. So now we move on to our next special somatic afferent. And this special somatic afferent is for the sense of hearing and balance, or the sense of acoustic sense and balance. And it is done with the help of the organ called ear. So this diagram gives you an overall view of the organs involved. We have three parts of the ear if we talk anatomically. One is called as external ear. External ear is made up of this ear cartilage or this large standing dome-shaped structure, which is called as pinna. And the external ear also consists of this narrow passage, which is called as external ear canal or the auditory canal. It has a vertical portion and it has a horizontal portion in dogs. And this canal ends at the eardrum or the tympanic membrane. So this whole space, the external ear canal or external acoustic meatus, as well as the ear cartilage or external ear pinna, these two structures, they make the external ear of the animal. Then we have the middle ear. This middle ear is beyond the tympanic cavity, and this small place which is present in the tympanic part of the temporal bone, this part. This small cavity or the middle ear cavity, it is also connected with the pharynx of the animal through the auditory or the eustachian tube. Then comes the petrous part of the temporal bone. And this petrous part has a small cavity inside which houses the inner ear, as we can see here. So I repeat here that the ear has three portions. The first one is external ear and is composed of pinna and external acoustic meatus or external auditory canal. Then the second portion is called as the middle ear, and it is still here. This part is not part of the middle ear. So this is your middle ear, and it is present in the temporal part of the temporal bone, tympanic part of the temporal bone. And we call it as the middle ear. This middle ear, through a canal or a tube-like passage, which is called as auditory or eustachian tube, is connected with the pharynx of the animal. Then we have the third or the inner ear. This inner ear of the animal is located within the petrous part of the temporal bone, and it is made up of two structures. One is cochlea, which is for hearing, and one is the vestibule or the semicircular canals, which is for the balance. So here we can look into more detail of the middle ear. Here we have this middle ear cavity, this small space. And if you see, this is tympanic membrane. Tympanic membrane is attached to the ear bone, which is called as malleus. Malleus is articulating or making joint with the next ear bone or ossicle. We call it as incus. And the incus is articulating with the third ear bone, which is called as stapes. So together these three are called as ear ossicles, starting from the malleus till the stapes. And the stapes is now like a handle attached to the small opening in the petrous part of the temporal bone. So it is basically covering that opening. So this is how your external ear through these three bones is communicating with the inner ear. What happens is that when you have the sound waves striking with the eardrum, it makes the drum vibrate. When the drum vibrates or the membrane vibrates, it vibrates the three bones. And this, when the stapes is vibrating at this small window-like opening, it is transferring the vibration into the fluid which is present in the inner ear structures. And when that fluid vibrates, that mechanical energy is basically then converted by the receptors or the neurosensory epithelium into the electrical energy. This is how sound is produced. The mechanical vibrations which are there in the fluid of the inner ear, they are basically stimulating the neurosensory epithelium to generate electric impulse, and that impulse is then taken as the sound or the sense of balance that you have. So this much for the middle ear, and then we will look into the structures of the inner ear. So if we look at the structures of the inner ear, we said that we have two different components. One is the spiral cochlea, and the epithelium in the spiral cochlea has the neurosensory component, which gives rise to the axons, which are later on forming the cochlear nerve, and that is for the sense of hearing. And similarly, this vestibule has three semicircular canals, and these three semicircular canals are at right angle to each other. You can think of the physics X, Y, and Z axes. So these three canals are at those X, Y, and Z axes, and as the animal's head turns this or that way, the changing axis of these three semicircular canal, it creates a nerve impulse which informs the animal of its changed orientation. So that is how it helps the animal with its balance. So the nerve which is originating from the epithelium of the semicircular canal is called as the vestibular nerve, and the vestibular and the cochlear nerve together, they form the eighth cranial nerve, and they are then helping the animal with its balance and hearing. So this much for the special somatic afferent for the sense of hearing and balance. Now we move on to the second last learning outcome about the structures involved in the sense of taste. It is also called as sense of gustation. And just like other senses, we have certain cells and certain structures which help the animal detect the taste. And we'll see how those structures are distributed in the epithelium. So these gustatory receptors or the gustatory cells, they are basically located on the surface of the tongue. And on the surface of the tongue of the animal, there are certain raised structures. These raised structures are called as papillae. And among those papillae, some of the papillae are specialized for detecting taste, and we call them as gustatory papillae. Here we are seeing one example of the gustatory papillae, which is velate papilla. And the velate papilla is like a mushroom. And if you see that this papilla is surrounded by the surface of the tongue, which is around it by a deep pit. This deep pit, which is there, it has the neurosensory epithelium in it. If you see the enlarged view of this pit here, and you see these small blue fusiform structure, so one of them is enlarged here. So this is the pit, the surface of the pit, which is this surface of the pit. So this surface of the pit has a small opening into it, and this small opening allows the food or whatever the solution or is there to enter this small opening. And then the cilia, which are here, which are the tips of the neurosensory epithelium, they are then sensing the chemical composition of this fluid, which is called as, this opening is called as the taste pore. So through the taste pore, the fluid which is present in the oral cavity, or the food or whatever particles which are there, which are dissolved in the saliva, they are entering into this taste pore. And here we have the cilia of the gustatory cells. We call them as gustatory hair. And they are then sensing the chemical composition of that fluid. And that chemical composition, then the sense by the epithelia is then transduced, as we say, they are acting as transducer. So they are here converting that chemical composition information, and that chemical composition is leading to generation of an impulse, and that impulse is taken up by the axons of the neurosensory epithelium, and these axons together, they make the nerves which are responsible for detection of sense of taste. And if you remember from the very early in this lecture, we said those nerves are the 7th and 9th nerve, that is facial and glossopharyngeal nerves. So I repeat here that the sense of taste is picked up by the taste buds, and these taste buds are basically present there in the form of gustatory papilla. And if you see here, the gustatory papilla has a pit outside it, and in this pit are the neurosensory epithelium cells. And one of the taste bud here, which we are saying is present in this pit, this taste bud is enlarged here. And if you look at this taste bud, it is made up of multiple cells. Some of them are basal cells, some of them are neurosensory epithelium cells, and some of them are plain supportive or sustentacular cells. So the cells which have neurosensory function, their tips are associated with hair-like structures or the cilia, and these cilia are picking up the chemical composition of the food which is there on the surface of this epithelium or the surface of the tongue. And this chemical composition leads to generation of an electrical impulse by the taste bud, and that electrical impulse is taken away by the help of sensory nerves, which are the, for us, special visceral afferents, and they are present here in the facial as well as glossopharyngeal nerve. So different animals have different type of gustatory papilla on them. And this would be your work or assignment to look into the Koenig and Liebig, or you can look into your Pascini or Dice and see which papilla are present in which animal. I'm just reading a few of their name. You will see that this fungiform papilla would be there. You would see that velate papilla would be there. Then foliate papilla would be there. So, and fungiform is again here. So you have to look into different animals' anatomy of the tongue and see which type of papilla are there. And you will see that among these papilla, fungiform, velate, and foliate, They are the gustatory papilla. That means that these papilla have taste buds on them. You will also come across certain papilla on the surface of the tongue of the animal, which are only mechanical papilla, which means that they do not have any taste buds with them. So you have to read some information about it from your textbooks before you come for the session on Monday. So that finishes our second last learning outcome. And now we'll move on to the last one, which is also special visceral afferent. But this is the sense of olfaction or sense of smell. So if you look at this diagram, these chemical substances or molecules, which are representing a certain smell, they are entering into the nostrils of the animal and then the nasal cavity. And here in the caudal part of the nasal cavity is the olfactory epithelium, which senses the chemical composition, and that leads to generation of an electric impulse in the neurons. And then this sense of olfaction is then processed into the brain. So we will see which structures are involved from the tip of the nose till the olfactory area of the brain and how it happens. On the left-hand side, we have an image. So first I would just ask you to look at the image below. And it says that in the face or the facial part of a dog, the cranial or the rostral part of the nasal cavity, that has typical respiratory epithelium. But the caudal part of the nasal cavity, the epithelium is called as olfactory epithelium or neurosensory epithelium. And there are cells dispersed in this epithelium which give rise to olfactory nerve axons. And immediately behind the nasal cavity and in the cranial cavity, we have the olfactory bulb. They look very close together here, but you have to realize that the nasal cavity is separated by the cranial cavity by a bone which is called as ethmoid bone. So the neurons or the axons of the olfactory nerve have to pass through the ethmoid bone to reach this olfactory bulb, where the initial processing takes place, and the olfactory bulb eventually then goes into the cortical area. The diagram above this one, it shows an image, which is a scan, and it again highlights the same structures. Here we have the rostral part of the nasal cavity, which is the respiratory epithelium. Then we have the caudal part, which has olfactory epithelium. Then the nerves pass through the ethmoid plate into the cranial cavity, where we have the olfactory bulb beginning and forming the whole tract. So here the same structure is shown in the middle picture, that the nasal cavity inhales the air which has certain smell into it, which could be a perfume, which could be food or anything. And then here we have olfactory epithelium. The olfactory epithelium leads to the olfactory nerve, which passes through the ethmoid bone and enters into the olfactory bulb, which leads to olfactory tract and then eventually the cortex. Now, if you enlarge this image, it scientifically or histologically, you can identify these structures. Let's start from this epithelium. So here we have the olfactory epithelium, which is made up of small basal cells, then tall sustentacular or supporting cells, and in between the basal and sustentacular cells are the neurosensory epithelium cells, which are basically nervous tissue. And the tip of these cells have cilia, which are embedded in the nasal mucus, and they are sensitive to the composition of this air. So the molecules which are representing a certain smell, they stimulate these neurosensory epithelium cells. That results in generation of an impulse, and this impulse is then carried by all these axons together, which are making the nerve fibers of the olfactory nerve. So these axons are then passing through the ethmoid bone through small holes which are present in this bone, and they enter into the olfactory bulb, which is an enlarged structure. And from there, the olfactory tract is formed, which eventually leads into the brain. So this is how the sense of smell operates in the animals, starting from the olfactory epithelium to the nerve fibers of the olfactory nerve, through the ethmoid plate into the olfactory bulb, the olfactory tract, and the brain. But in animals, we have another very important structure, the vomeronasal organ associated with the sense of olfaction. This vomeronasal organ is sensitive to the pheromones, and this pheromones are those chemical substances which, when an animal smells, it leads to the modification of their behavior. It is an important part of the sexual behavior of the animals also. But where it is located, if you look into this diagram here, this is the location of the vomeronasal organ, which is present in the hard palate of the animal, and it opens into the oral cavity. So you might have seen certain dogs which are curling their upper lips. So they open their oral cavity to expose the vomeronasal organ to the air or to the pheromones. And this vomeronasal organ has sensory fibers or axons leaving it, and they also join the olfactory nerve and then the same route. So this is an extra area or an extra you can say structure in animals which is associated with the sense of smell, but particularly for the pheromones. And it allows the animal to display its behavior, which is required for mating and for detecting the other animal's presence there. It is also called as Jacobson organ or apparatus. Here is an MRI or medical resonance, magnetic resonance image of a dog's skull. And this view shows you, this VNO is vomeronasal organ. This view shows you the location right on either side of the median plane in the palatine bone. Here we have the vomeronasal organs on each side. This is a cross section, and here you see this vomeronasal organ. One is here and the other is here. And this is a side view. And in the side view, you can see the vomeronasal organ here embedded in the hard palate. So it has an opening here, and through the oral cavity, it detects or gets the maximum amount of air that can it get to sample the smell. And then the axons which are leaving this vomeronasal organ, they join the olfactory epithelium here. So this is how the dogs, in addition to their regular olfactory epithelium, get extra sense of the pheromones through the vomeronasal organs. So this much about the receptors and proprioceptors, exteroceptors, interoceptors, whatever term you used for them. And I would just say that the afferent system that we have in the animal. So go through this recording, I would say 1 or 2 times, and prepare your list of questions that you want to ask me. And we'll then have a Q&A session on Monday for A and B groups and on Thursday for C and D groups.